Hi everyone, today we are going to talk about Jellyfish Part 2. We are going to talk from where we left off. If you don't know the Part 1, go ahead and watch it. The study of jellyfish eye evolution is an intermediary to a better understanding of how visual systems evolved on Earth. Jellyfish exhibit immense variation in visual systems, ranging from photoreceptive cell patches seen in simple photoreceptive systems to more derived complex size seen in box jellyfish. Major topics of jellyfish visual system research: the eye, morphology and molecular structures of box jellyfish, and various uses of vision, including task-guided behaviors and niche specialization. Experimental evidence for photosensitivity and photoreception in cnidarians antecedes the mid-1900s, and a rich body of research has since covered evolution of visual systems in jellyfish. Jellyfish visual systems range from simple photoreceptive cells to complex image-forming eyes. More ancestral visual systems incorporate extraocular vision that encompasses numerous receptors dedicated to single-function behaviors. More derived visual systems comprise perception that is capable of multitask-guided behaviors. Although they lack a true brain, cnidarian jellyfish have a ring nervous systems that plays a significant role in motor and sensory activity. This net of nerves is responsible for muscle contraction and movement and culminates the emergence of photosensitive structures. Across cnidaria, there is large variation in the systems that underlie photosensitivity. Photosensitive structures range from non-specialized groups of cells to more conventional eyes similar to those of vertebrates. The general evolutionary steps to develop complex vision include non-directional photoreception, directional photoreception, low resolution vision, and high resolution vision. Increased habitat and task complexity has favored the high resolution visual systems common in derived cnidarians such as box jellyfish. Basal visual systems observed in various cnidarians exhibit photosensitivity representative of a single task or behavior. Extraocular photoreception is the most basic form of light sensitivity and guides a variety of behaviors among cnidarians. It can function to regulate circadian rhythms and other light-guided behaviors. Among cnidarians, it can function to regulate circadian rhythm and other light-guided behaviors responsive to the intensity and spectrum of light. Extraocular photoreception can function additionally in positive phototaxis, as well in avoiding harmful amounts of UV radiation via negative phototaxis. Directional photoreception allows for more complex phototactic responses to light and likely evolved by means of membrane stacking. The resulting behavioral responses can range from guided spawning events timed by moonlight to shadow responses for potential predator avoidance. Light-guided behaviors are observed in numerous cyphozoans, including the common moon jelly Aurelia aurita, which migrates in response to changes in ambient light and solar position, even though they lack proper eyes. The low-resolution visual systems of box jellyfish is more derived than directional photoreception, and thus box jellyfish vision represents the most basic form of true vision, in which multiple directional photoreceptors combine to create the first imaging and spatial resolution. This is very different from the high-resolution vision that is observed in camera or compound eyes of vertebrates and cephalopods that rely on focusing optics. Critically, the visual systems of box jellyfish are responsible for guiding multiple tasks or behaviors, in contrast to less derived visual systems in other jellyfish that guide single behavioral functions. These behaviors include phototaxis based on sunlight or shadows, obstacle avoidance, and control of swim pulse rate. Box jellyfish possess proper eyes that allow them to inhabit environments that lesser derived medusa cannot. In fact, they are considered the only class in the clade Medusozoa that have behaviors necessitating spatial resolution and genuine vision. However, the lens in their eyes are more functionally similar to cup eyes exhibited in low-resolution organisms and have very little to no focusing capability. The lack of the ability to focus is due to the focal length exceeding the distance to the retina, thus generating affected images and limiting spatial resolution. The visual system is still sufficient for box jellyfish to produce an image to help with tasks such as object avoidance. Box jellyfish eyes are a visual system that is sophisticated in numerous ways. These intricacies include considerable variation within the morphology of box jellyfish's eyes and the molecular makeup of their eyes, including photoreceptors, opsins, lenses, and synapses. The comparison of these attributes to more derived visual systems can allow for a further understanding of how the evolution of more derived visual systems may have occurred, and puts into perspective how box jellyfish can play the role as an evolutionary model for all visual systems. Box jellyfish visual systems are both diverse and complex, comprising multiple photo systems. There is likely considerable variation in visual properties between species of box jellyfish. Given the significant interspecies morphological and physiological variation, eyes tend to differ in size and shape along with number of receptors and physiology across species of box jellyfish. Box jellyfish have a series of intricate lensed eyes that are similar to those of more derived multicellular organisms such as vertebrates. Their 24 eyes fit into 4 different morphological categories. These categories consist of 2 large morphological morphologically different medial eyes containing spherical lenses, a lateral pair of pigment slit eyes, and a lateral pair of pigment pit eyes. The eyes are situated on rhoophalia, which serve sensory functions of the box jellyfish and arise from the cavities of the exumbrella on the side of the bells of the jellyfish. The two large eyes are located on the midline of the club and are considered complex because they contain lenses. The four remaining eyes lie laterally on either side of each rhoophalia and are considered simple. The simple eyes have observed as small invaginated cups of epithelium that have developed pigmentation. The larger of the complex size contains a cellular cornea created by monocylated epithelium. Cellular lens, homogeneous capsules to the lens, vitreous body with prismatic elements, and a retina of pigmented cells. The smaller of the complex eyes is said to be slightly less complex given that it lacks a capsule but otherwise contains the same structure as the larger eye. Box jellyfish have multiple photosystems that comprise different sets of eyes. Evidence includes immunocytochemical and molecular data that shows photopigment differences among the different morphological eye types, and physiological experiments done on box jellyfish suggest behavioral difference among photosystems. Each individual eye type constitutes photosystems that work collectively to control visually guided behaviors. Box jellyfish eyes have multiple photosystems that comprise different sets of eyes. Box jellyfish eyes primarily use CPRCs, ciliary photoreceptor cells, similar to that of vertebrate eyes. These cells undergo phototransduction cascades that are triggered by opsins. Available opsin sequences suggest that there are two types of opsins possessed by all cnidarians, including an ancient phylogenetic opsin and a sister ciliary opsin to the C-opsins group. Box jellyfish could have both ciliary and cnidops, which is something not previously believed to appear in the same retina. Nevertheless, it is not entirely evident whether cnidarians possess multiple opsins that are capable of having distinctive spectral sensitivities. Comparative search on genetic and molecular makeup of box jellyfish's eyes versus more derived eyes seen in vertebrates and cephalopods focus on lenses and crystalline composition, synapses, and PAX genes, and their implied evidence for shared primordial genes in eye evolution. Box jellyfish eyes are to be an evolutionary model of all eyes based on their evolutionary recruitment of crystallines and PAX genes. Research done on box jellyfish, including Trepidalia cystophora, has suggested that they possess a single PAX gene. PAX B functions by binding to crystalline promoters and activating them. PAX-B in situ hybridation results in PAX-B expression in the lens retina and stereocytes. This results in the rejection of the prior hypothesis that PAX6 was an ancestral PAX gene in eyes has led to the conclusion that PAX-B was a primordial gene in eye evolution, and that the eyes of all organisms likely share a common ancestor. The lens structure of box jellyfish appear very similar to those of other organisms, but the crystalline and distinct in both function and appearance. Weak reactions were seen within the sera, and there were very weak similarities within the crystallines among vertebrate and invertebrate lenses. This is likely due to difference in lower molecular weight proteins and the subsequent lack of immunological reactions with anti-sera that other organisms' lenses exhibit. All four of the visual systems of box jellyfish species investigated with detail have invaginated synapses, but only in the upper and lower lensed eyes. Different densities were found between the upper and the lower lenses and between species. 4 types of chemical synapses have been discovered within the rofaglia, which could help in understanding neural organization, including clear unidirectional dense core, unidirectional, clear bidirectional, and clear and dense core bidirectional. The synapses of the lensed eyes could be useful as markers to learn more about the neural circuit box jellyfish retinal areas. This was all. Bye, and I'm going to do a part 3. Bye.